Well, thanks so much for giving me the opportunity to be here today and be on this panel. Access and inclusion is an important consideration as we think about the ways of having more innovative trial design. I'm going to try clicking again and see if I get queued up. All right, so I'll wing it for a moment. One of the things when I was asked to be on this panel gave me pause, frankly, because Patients Like Me is an online platform. We have already recognized long ago that we will not be as representative of the general population as other environments might be that don't depend on this sort of technology that we have to have people able to use. One of the things that we try to do, no matter how we're thinking about engaging with people across different conditions, is to really think about how can we reach out in a different way. If we're working in lupus, for example, which we've been doing now for a couple of years, we've spent a lot of time and thought and thinking about how do we best identify areas where we can work with a community of African American women, for example, who are disproportionately represented in that particular disease state, because we know that we won't typically draw them into our environment without some specific outreach. So that is one area where we will spend some time. What I decided to focus on, because I didn't feel that I would really be able to speak to the access and inclusion issue as clearly as someone who is working with a more underrepresented population, was to focus on one condition and to give you a few examples of how we have been able to work with virtual trial design or patient-initiated trials to actually include people who typically would not have been a participant in a research trial because of the exclusion criteria associated with their condition. And that condition is ALS. And for those of you who are unfamiliar with Patients Like Me, we were founded by a family that actually had an experience with ALS in Stephen Haywood, who was a 29-year-old man diagnosed with it, and his brothers and family really marshaled their energies to identify ways of being able to learn as much as they could, but recognize that much of what they were learning was from other people who were either living with ALS or actually were caregivers of ALS. And so the impetus behind Patients Like Me really emanated from this desire to have more access to data that actually could be coming from the real world, from real people living with a real condition. So thank you very much, and I'll get started now with the slides. But that was a little bit of a prelude why I chose this condition. We have been working in this area for over 14 years, and we have some interesting insights that I think might be useful for this discussion. So I'm going to talk about three areas. One is patient-initiated research in ALS, and there's two areas there I'll cover. Lithium carbonate study we did a while back, and then a group called ALSUntangled, and then a hybrid trial that we have completed, and that was done with the ALS clinic at Duke, and then two that are now currently in the planning stages, also going to be done with Duke's ALS clinic. So we'll get started. Now, many of you in the room who I know pretty well are probably familiar with the lithium carbonate study that we did, and you're probably tired of hearing about it. But I think it sets the stage today for being able to say 10 years ago, we had patients come to us as a result of learning about a small study done in Italy that showed among 16 patients that lithium carbonate was effective in slowing the progression of ALS. As you might imagine, in this fatal disease, people who have ALS were quickly looking to get their physicians to prescribe them lithium. But at the same time, they were also being quite empowered to say, you know, we need to monitor ourselves in the same way people were monitored in this trial to be able to determine whether or not we can get the same results. So they recognized the absolute need to have data that could help them better understand what the experience was. So because we had already begun working in this area and we had already started with some specialty in ALS, we actually worked with the patients to create the design. We actually were able to recruit 160 people pretty quickly, which was 10 times the number of people that were in the original study. But importantly, one of the things that we did here was actually work looking at all of the data that we already had about people with ALS and their progression scores using a clinical endpoint that's used in clinical trials, the ALS functional rating score, to see if we could create an algorithmically driven control cohort. So finding ways of being able to find progression curves that mapped to the people who were actually on drug to see how close we were getting to whether or not what they were experiencing enhancing was traditionally what one would expect to see in people with a similar type of progression. So that matching algorithm actually has been refined and published. All of the data that we use to create that has been made public so everyone could to give us that peer review feedback. And it's actually part of the mechanisms that we use currently for the new trials that we're developing. So we always go back and try to identify a matching cohort for controls that would typically be able to then do an analysis of what their progression looks like against those who are actually participating in the virtual trial. So the result of this actually was within 6 months, we were able to refute the findings of the clinical trial. And it took a few years later for a number of NIH studies to come to the same conclusion. But fast forward a number of years, about a year and a half ago, there was a rather comprehensive meta-analysis of all the ALS studies that involved genomic data. And what the researchers identified was a genetic variant that actually was in a small subset of people with ALS, less than 20% of the people, that actually was getting washed out in the analysis that was being done because there were so few of them. What they identified was people with this variant called UNC13A were actually having a response to lithium, even albeit small, but there was some indication that there should be further study in this area for that particular group of people. So we are underway with the researchers in the UK and the Netherlands to start standing up a study where we will actually begin to look at identifying people with this variant and then determining whether or not there might be some promise for using a lithium carbonate. But what I like about this is that we didn't know this information 10 years ago. We know this information now. It's time for us to then take what we learned and then start applying it now with new data, and new sources of information that we wouldn't have had access to before. So I also wanted to introduce you to something called ALSUntangled. So about the same time that we were doing our work, ALSUntangled was founded, and it was a consortium of patients, clinicians, and researchers that really are committed to understanding what's the impact of the alternative off-label treatments that people in ALS often turn to for whatever reasons. They may have seen it in an infomercial, they may have seen it online somewhere, but there's not enough evidence for anyone to really be able to say that this might be something worth trying. So the ALSUntangled Consortium, of which we are a member, actually it represents global representation, and what it does is it seeks to identify things from patients themselves who report certain things about nutritional supplements, other sorts of complementary or alternative modalities that are often being used off-label, and puts them into a queue for voting to determine which of these might we want to start exploring. So the patients actually rate those based on some of the information that they're gathering, but then the scientists come together with the patients and they start to do what really gives an analysis of the potential of that particular product to have any sort of validity or opportunity to explore further. So they've developed this table of effectiveness, and I'm just showing you a piece of it here, but the reference here will take you to the publication about it. And so everything that comes up to be considered for inclusion in doing some further study is judged against these particular areas. So the mechanism of action, is it relevant to ALS? Is there some reason to believe that theoretically this might work in ALS? Have there been any preclinical models that we can look at that could tell us more about this particular product? Are there any case reports from patients themselves? Have there been any trials at all? And if there have they been peer-reviewed? Do we have more information and literature on them? And what are the identified risks that are known about this particular product? And it's gauged against U being unknown, so that could be something that any one of these things might have a U. But then it goes on a grading system of A to F. And just like we always had in school, A to F generally tells us that A looks pretty good and F doesn't look good at all. So with every product that you see on here, and there's been 40 of them done in full reviews to determine whether or not this has promise and whether it should actually go forth. Two of them I'm going to point out because I'm going to talk to you about those in a minute. One of them is Lunasin, and it's a soy peptide that actually has been found in the literature. It has some theoretical basis for believing that it could be relevant for ALS. There's case studies from patients. There has been a peer-reviewed trial with it, and there's some additional information around its risk being relatively low. So it was an area that the Duke ALS Clinic and Patients Like Me decided we would explore more further. And then curcumin, which is another product that's actually typically found in things like curry and some other products is something that, again, has some theoretical value to the ALS population. And you can see it also has C's and B's, where the oral formulation of this gets a B for its risk, where the IV formulation gets a D. So those are the ways that we would then begin to think about how we might structure a virtual trial using these particular products. So I'm going to talk to you first about the Lunasin study. So this was what we called a hybrid virtual trial, and I'm certainly open to coming up with any other names that we might come away with from this meeting. But this particular trial required patients to actually have clinic visits on day 1, day 30, and day 365. So all other times they were actually putting information into their Patients Like Me profile. They were also being contacted by the clinic nurse, and they were also being contacted by our community moderator. So there was a lot of touch with the participants that were involved in this. It was a small study. We had 50 patients, but we set out to only recruit 50 patients. We wanted to determine feasibility. We wanted to better understand whether this type of model could work. We were able to recruit about 10 people a month, so it was recruited and fully recruited, enrolled in a relatively short period of time. So the opportunity here was really to also measure whether or not Lunasin showed any particular improvement in the progression scores, which it did not. But actually, how did the patients and the participants in this study feel about engaging in a study that didn't require so much involvement of being in a clinic setting most of the time that they would be involved? The other thing we wanted to determine is, could we increase access and inclusion by doing something that didn't necessarily follow the usual clinical trial requirements for ALS studies, which typically require a person to have a disease no more than 36 months, and also to have relatively high scores for respiratory function as well as swallowing function. And what we found was when we eliminated those particular requirements, we were able to get a more representative subset of people that would participate, people who were on average about 5 years with disease, some of them 13, the range was from half a year to 13 years, so those people beyond the 36-month criteria would never have been included in a trial. And we also found that the participants oftentimes had some respiratory support, whether it was a non-invasive ventilator or some other support that they needed, as well as a feeding tube. And again, these would be exclusion criteria for most of the studies in ALS today. Our retention rate was about 89%, so people were satisfied with it and stayed with it and were committed to being a participant in this. So we feel like we actually were able to show that this model actually is really quite effective when you're trying to engage people, especially in a condition for which participating in a trial can be very challenging, or for which they may never be able to get enrolled in because of the various criteria that are typically involved. So in terms of access and inclusion, we feel this was a model that we could then start to use as an exemplar going forward in other conditions that we would work in. And then finally, let me tell you a little about a couple that are in the planning stages. We have felt pretty positive in our relationship with the Duke ALS clinic. We've done some really good, exciting work with them. And so now we're working on 2 areas. Both of these have gone through the process of the ALSUntangled methodologies, just so we would get a better sense of using things that traditionally would have gone through that process. And the trial design is being built on what our experience has been with the Lunasin trial. So both of the studies include a range of different type of patient-reported data, as well as blood specimen collections that we're getting, and we actually send a phlebotomist into the home for that collection, so the patients don't have to go into the clinic for that. So ALS reversals is an interesting area where there have been some very isolated and limited case studies reported of people using a particular product that actually had a reversal of symptoms, not a reversal of complete disease, but a reversal of symptoms that actually went back to a point that they could point to say, this is how I was 8 or 9 months ago, and yet now that it was not something that, the only thing that changed was I was using this particular supplemental product. So that initiation by patients themselves coming to the ALSUntangled group sort of prompted the interest in doing a curcumin study, because that was the product that was reported. So we are just getting ready now to actually launch 2 of these projects. One will be just looking at people who have reported ALS reversals or some indication of them, to see if we can, first of all, identify anything unique about them them in the patient-reported data, but also in their biological data. We will be doing some specimen collections on these as well. And then the curcumin trial will actually be completely virtual. There will be no requirements for going into the clinic on these other than the initial enrollment, which will happen at the Duke ALS clinic site. And then from there, all of the interaction will be providing the Duke clinic will provide the drug via the delivery to the home, and we will be monitoring them through our usual platform as well as through blood collection that will be done at different points in time throughout the course of the trial. So I'm going to end there, but I'm hoping my message is clear that in a particular condition that typically is pretty limited in how people can access their participation in research because of the rapidity of the disease and the sense of that these patients oftentimes lose the functions one would have to have in order to participate in a usual trial. This type of environment allows itself for people to participate even when their functioning is well assisted by different devices that we would typically not be able to include those in our usual studies. And the last thing I'll say is that we had a patient-initiated study done a few years ago where we actually extended the ALS-FRS rating score to include 3 additional items. This particular person was a professor at Brown. She'd been living with ALS for many years, and she was excluded from every trial she applied for because she scored 0 on that score. Now, 0 on that score from a clinical perspective means you're dead. And like the Monty Python movie, she says, I am not dead yet. I am still quite functional, even though everything I do is assisted by a device, breathing and all the other things that she would do. So what we were able to do was to create 3 new items for that score that are actually have now been used in some NIH studies where the person's cognitive ability is assessed in terms of their inclusion criteria, their ability to be able to use some computer-assisted device, whether it's through eye measure, and then a few other ways of being able to assess their capacity to be able to engage in the trial through either their language criteria and things like that. So, but those 3 additional extensions are now part of the ALS functional rating score as a result of a patient initiating that activity, and she is a co-author with us on that publication. So I'll stop there. Thank you.